Hi, I'm David and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. In figure 1.1, we have a photomicrograph of a chloroplast. Question A wants you to complete the table by identifying the letter in the figure that is a location matching the description. Let's identify the parts represented by each of the letters. A is the fluid surrounding the grana, so it is the stroma. B points to the space enclosed by the thylakoid. It is called lumen. C is the thylakoid membrane. D is a ribosome. Chloroplast has 70 S ribosomes in its stroma. In cyclic and non-cyclic photophosphorylation, the passing of electrons in ETC occurs on the thylakoid membrane. Protons are being pumped from the stroma into the lumen of the thylakoid. So, the answer is B. Triose phosphate is made in the Calvin cycle that takes place in the stroma. The ribosome is the site of protein synthesis. So, D is where some chloroplast proteins are made. Protons are pumped by the electron carriers on the thylakoid membrane. These electron carriers form the ETC. B1 wants you to describe how to carry out chromatography. There are two things you must mention in your answer: the procedure to separate the pigments and how to identify them. Let's start with the procedure for the separation of the pigments. First, you have to place a spot of the extract on the pencil line at one end of the filter paper. You can also call it a baseline or the line of origin. We usually repeat this process to concentrate the spots. This is to ensure the chromatogram produced to be clearer. This end of the paper is suspended in the solvent without the solvent touching the spot, as the spot may dissolve in it. Cover the gas jar with a lid to prevent the evaporation of the solvent. Many organic solvents have a low latent heat of vaporization. They evaporate quickly. The solvent will start to move up the filter paper. Before the solvent overshoots, remove the paper and mark the solvent front. The solvent front is the furthest distance the solvent traveled on the filter paper. To identify the pigments, RF values are calculated. RF value is the distance moved by a pigment divided by the distance moved by the solvent front. When the RF value of each pigment is obtained, we can now compare these RF values with non-RF values to identify them. We can do this because the RF value of a pigment is fixed when a specific solvent is used. B2. Explain why membrane C has many different colored pigments to function efficiently. The role of these pigments is to absorb light energy. However, different pigments have peak absorption at different wavelengths. Having different colored pigments can increase the range of different wavelengths to be absorbed. More light energy is available for photosynthesis. So, the rate of photosynthesis, particularly the light-dependent reaction, increases. In other words, this increases the action spectrum. You can also get a mark by naming at least two different pigments. In question 2, we have a passage about the Hawadak Island in Alaska. You are asked to define ecosystem and niche. A common mistake when defining an ecosystem is that many students think an ecosystem refers to all living things in a defined area. That is the definition of community. An ecosystem is a self-contained unit containing a community of different organisms or species and their interactions with biotic and abiotic factors. In other words, these organisms of different species interact with each other and with their physical environment. Niche is the role or function of a species within its ecosystem or habitat. For example, a fruit tree provides shelter for insects and food for birds in a habitat. B describes a method ecologists use to measure the abundance of invertebrates and seaweeds on the island. 1 wants you to name the sampling method. According to the description, tapes were laid on the site. Then, quadrats were placed at 5-meter intervals next to the tapes. This is a transect, as a tape line is established to sample an area. To be more specific, it is an interrupted belt transect. Interrupted refers to the fixed interval in which the sampling is done. It was not carried out along the whole line. It is a belt transect, as quadrats were used to provide a sampling area, so it is not just a line transect. B2 wants you to identify the levels of biodiversity that were assessed and not assessed. The three levels of biodiversity are ecosystem or habitat diversity, species diversity, and genetic diversity. The abundance of species was determined in the sampling method. It was done by measuring the percentage cover of those species. It is a part of species diversity. The other two were not measured in the research. Table 2.1 shows the percentage change in abundance of some organisms on the island. C1. State the genus name of one organism that has increased in abundance. Both the black oystercatcher and glaucous winged gull increase. Their scientific names are included in the table. The first word of the name is the genus, while the second is the species. So, Hematopus and Larus can be used as the answer. 2. Outline the features of the kingdom Protoctista that are shown by the seaweeds. Seaweed is a type of algae. They are eukaryotic. Algae can carry out photosynthesis. They are autotrophs as they can synthesize their own organic compounds. Like plant cells, algae have cellulose cell walls. However, do remember that seaweeds are not plants. They are aquatic organisms that are commonly found in the ocean as well as in the rivers, lakes, and other water bodies. Algae are not motile. You can use the word sessile to describe this. It means they are permanently attached or fixed and not free-moving. They are multicellular organisms. Unlike plants, algae do not have vascular tissue such as xylem and phloem. In D, there is a table that shows the percentage differences in the abundance of organisms in rat-free islands compared with rat-occupied islands. Suggest and explain how removing rats has restored the ecosystem on Hawadaks Island. The command words are suggest and explain. So, you should not just state the differences and quote the data. You need to provide reasons for the changes to gain full credit. The table shows that both species of birds increase. This is because the rats are the predators of their eggs or chicks. If the birds increase, they will consume more mussels and sea snails, so their number decrease. Mussel and sea snails feed on seaweed, so their decrease will cause an increase in the abundance of seaweed. These 3 points are the explanations for the changes using the food chain. When you compare this table with table 2.1, you will notice that Hawadaks Island is getting more and more similar to a rat-free island. This indicates the biodiversity of Hawadaks has been restored to the state before rats appear on the island. Lastly, mentioning that the rat is an invasive or alien species would get you a mark. Invasive species are those non-native plants and animals that threaten native wildlife and ecosystem. They are not from the area and may cause ecological havoc. Question 3 is about rice plants. A1. State the function of parenchyma. Parenchyma is a soft plant tissue containing many air spaces. Those air spaces provide a low-resistance internal pathway for the easy movement of oxygen in the plant. It is important for them as the root part of a rice plant is submerged in the water. It is difficult for the cells to get enough oxygen. Parenchyma enables air to be supplied to the submerged root cells. With sufficient oxygen, the cells can carry out aerobic respiration, which produces more ATP compared to anaerobic respiration. 2. State one other way in which the roots of rice are adapted to being submerged in water. Note that you can only use the adaptations related to the roots as your answer. The cells can carry out anaerobic respiration in the form of ethanol fermentation. Anaerobic respiration allows glucose to be oxidized in the absence of oxygen and ATP to be produced. However, ethanol, the product of anaerobic respiration, is toxic to cells if accumulates. So, rice plants develop a high tolerance to ethanol. They produce more ethanol for alcohol dehydrogenase, an enzyme that breaks down ethanol. 3. Describe how selective breeding could produce varieties of rice with fast-growing stems. Selective breeding is the practice of mating individuals with desired characteristics to increase the frequency of those traits in a population. To do this, farmers select those individuals with fast-growing stems and cross them. the offspring produced may or may not have the desired characteristics. So, they select and only breed the offspring with fast-growing stems. This process is repeated over many generations to produce a group with a high frequency of that trait. Outbreeding is carried out occasionally to avoid inbreeding depression. Inbreeding depression is the reduced survival and fertility of offspring due to the breeding of closely related individuals. 4. Explain how auxin affects the growth of rice stems. Auxin is a plant hormone or plant growth regulator. It increases stem growth. In other words, it stimulates a plant to increase in length or height. After auxin binds to the receptor on the cell surface membrane, it stimulates proton pumps to actively move protons into the cell walls. This causes a reduction of pH. An enzyme called expansin is activated. It catalyzes the breakage of the cross-links between cellulose microfibrils in the cell wall. With fewer hydrogen bonds, the cell wall becomes weaker. Water will then enter the cells by osmosis down the water potential gradient. The cell will elongate or expand. The increase in volume is now possible because of the weakened cell wall. B talks about the two types of wild rice plants and their characteristics. You are asked to explain how they may have evolved through sympatric speciation. Sympatric speciation is the arising of new species from another species while inhabiting the same geographic region. According to the information, Oryza nivara has a different feature from the typical rice plants. They are adapted to drier conditions or habitats. Because of this, the two groups are ecologically isolated. It means they grow and mate in different environments or area. The information also shows that they have different flowering times. This prevents interbreeding between them because when one species releases the pollens, the other one is not receptive. So, there is no gene flow between them. They are said to be reproductively isolated as they cannot reproduce with each other.
Polyploidy may have occurred. It is the condition in which a normally diploid cell or organism acquires one or more additional sets of chromosomes. When the chromosome numbers are different, they can't breed with each other. There is a mark for AVP. For example, you can say that polyploidy may be a result of an error in nuclear division.
4A1 wants you to define recombinant DNA. The word recombinant refers to the combination or joining of DNA from different organisms or sources. You can also say that the DNA are from different species. A2. Explain the advantages of using recombinant human insulin to treat people with diabetes. Since this technology uses microorganisms such as bacteria or yeast to produce insulin, it can be made in large quantities. Microorganisms have a fast growth rate. They can increase their number in a short time, and they can synthesize proteins rapidly. Secondly, it is cheap to make the protein. The procedure has a low cost or is more cost efficient compared to the traditional production method using animals. Since only microorganisms are used, this technique does not harm animals. In the conventional procedure, where proteins are extracted from animals or human carcasses, there is a chance of transferring infections from the animals to the patient. The use of recombinant human insulin does not have the same risks. The insulin gene inserted into the host cell has an identical sequence to the human insulin gene. So, it does not cause allergy, inflammation, or stimulate an immune response. Our body will not recognize the protein as non-self. Due to the identical sequence, recombinant insulin can fit better to the insulin receptors in the patient's body, so it acts more rapidly. Lastly, it is identical to insulin, which is present in human body all the time. The receiver does not develop tolerance for it. In B, we have the sequence of different types of insulin and the engineered insulin called analogs. 1. Identify the type of insulin that is most suitable for treating cats. The best insulin for cats would be the one most similar to a cat's insulin. Cow's insulin is the closest match. 2. Suggest ways in which analog insulin molecules can be produced by genetic engineering techniques. The table shows that the analogs have different amino acid sequences to human insulin. We can modify the nucleotide sequence of a human insulin gene to get the analog genes, and then use it to produce analog proteins. First, we should obtain the nucleotide sequence for the normal human insulin gene as a blueprint for the modification. Next, obtain insulin mRNA from a human cell. Then, use reverse transcriptase to synthesize human insulin. Reverse transcriptase is an enzyme that catalyzes DNA synthesis using RNA as the template.
Gene editing is used to alter the nucleotide sequence of the human gene. This new modified gene is the analog gene. The analog gene will then be inserted into a host cell such as bacteria or yeast. A promoter must be added so the gene can be expressed. A promoter is a region of DNA upstream of a gene where RNA polymerase and transcription factors bind to initiate the transcription of that gene. There is a mark for AVP. For example, you can provide some details for the steps mentioned in the main point above, such as the use of PCR to amplify the analog gene or the methods to introduce the analog gene into the host cells. C. Outline the advantages of using databases of nucleotide sequences to investigate evolutionary relationships between species. The study involves the analysis and comparison of a large quantity of information. By using the databases, we can share, search, and access data and information more quickly. A lot of data can be obtained in a shorter time. Besides, the data can be accessed wherever you are, as they are online. Comparisons can be done easily. Many software can be used to align the sequences next to each other and analyze all of your selected sequences simultaneously. Since all these do not need to be done manually, the process becomes much more rapid. The similarities and differences can be quantified too. Nucleotide differences can be counted and displayed within a short time. They can help us to determine how closely related two species are by counting the differences or mutations between the DNA we chose to compare. The fewer the differences, the closer they are.
Figure 5.1 shows a part of a nephron. A1 wants you to identify the structures labeled A and B. This is a bowl-shaped structure with a network of capillaries inside. So, this is a Bowman's capsule. The capillaries inside form a network called the glomerulus. The clear space in the figure is the capsular space where glomerular filtrate is found. 2. Describe how blood is filtered by the part of the kidney nephron shown in the figure. Since the question did not name the process, stating the name will give you the first mark. Ultrafiltration means filtration under pressure. The pressure here is hydrostatic pressure. The glomerulus has a high hydrostatic pressure due to the size difference between the afferent arteriole and the efferent arteriole. The wider afferent arteriole allows more blood to enter, and the narrower efferent arteriole allows less blood to leave, creating high pressure. The capillary endothelium has fenestrations. You can also call these gaps, holes, or pores. The fenestrations only allow small molecules such as ions, glucose, amino acids, and urea to leave the blood easily. They can easily escape the glomerulus, enter capsular space, and form the filtrate. However, there is a physical barrier between the endothelium and the podocyte of the Bowman's capsule. It is the basement membrane. It forms the main filtration so that not everything in the blood can enter the capsular space. Anything larger than a relative molecular mass of 68,000 to 70,000 is not passing through this barrier. Lastly, the inner layer of the Bowman's capsule is made up of podocytes. Due to their foot-like projections, slit pores are formed. This allows substances to pass through easily without crossing the cell surface membrane. Podocytes also synthesize the material to form a basement membrane. In B, we have a figure showing some steps involved in the cell signaling pathway, activated by glucagon. C is an enzyme activated by the G protein. It is adenylyl cyclase. You can also spell it as adenyl cyclase. it catalyzes the formation of the second messenger called CAMP from ATP. B2 talks about a condition called SIAD. Figure 5.3 shows how sodium ion concentration in the blood affects the ADH concentration in the blood of people with and without SIAD. You are asked to describe the results and explain the effect of type C SIAD on osmoregulation. First, make a general statement. As sodium ion concentration increases, ADH concentration increases in both cases. However, ADH rises in people with SIAD at a lower sodium ion concentration. You can get a mark by quoting comparative figures to support point number 2. The next few points are the explanation. The role of ADH is to cause more aquaporins to be added to the cell surface membrane of collecting duct cells. This leads to a greater permeability to water in the collecting duct. In SIAD, more ADH is released at a lower sodium ion concentration, so more water is reabsorbed at the collecting duct into the blood. This results in people with the condition have a smaller volume of more concentrated urine as the water content of the urine is lower. Since much water is reabsorbed into the blood, people with SIAD have a high blood water potential, and more water means a lower sodium ion concentration in the blood. Question 6 is about the inheritance of fur length and fur color of the domestic rabbit. Fur length is controlled by 2 alleles, while fur color is controlled by 3. A1 lists the 4 possible genotypes of the rabbit shown in figure 6.1. The rabbit has short fur, so it has at least one dominant allele for the fur length. Its fur has a Himalayan pattern. This is coded by the BH allele. Since it is dominant to the allele B, this rabbit can be homozygous BH or heterozygous with one BH allele. So, these are the possible genotypes. To produce offspring that only look like the parents with no other possible outcomes, the parents must be homozygous recessive. In this case, the genotype of a homozygous recessive rabbit would be a, small b. Its phenotype would be long fur and albino. Question B wants you to draw a genetic diagram. There are 2 crosses involved, but you only have to draw the second one. The first cross is between a homozygous long and black fur rabbit and a homozygous short white fur rabbit. All of the offspring produced have the same genotypes, big A, big B. We are now crossing 2 individuals of this generation. The gametes produced should contain one allele for each of the genes. In other words, every gamete must have one A and one B genes. There are 4 possible combinations. Then, use a Punnett square to show the possible outcomes when these gametes fuse. There are 16 different combinations. In the Punnett square, use brackets to indicate the phenotypes. Failure to do so may result in losing marks. Then, put them into the 4 categories and write the ratio. If you only write the ratio without stating which phenotypes they represent, you will not get this mark. C1. Explain why the 2 genes assort independently. Independent assortment occurs when the homologous chromosomes position themselves either way up on the equator of the spindle fiber. So, they segregate randomly. This happens to genes that are not found on the same chromosome. In other words, the genes are not linked. The bivalent or pair of homologous chromosomes orients themselves separately at the equator of the spindle. This is an event in metaphase 1 of meiosis. The random segregation of the chromosomes leads to 4 different combinations of the alleles in the gametes. The allele combinations in the parents may not be preserved in the next generation. This means the combinations of alleles in the offspring do not necessarily the same with the parental combinations. Table 6.1 shows a table used to calculate the chi-square value. In the genetic diagram we constructed in B, the ratio between the phenotypes should be 9:3:1. 48 individuals are being produced. You can get the expected values by multiplying the probability of each phenotype with the total. To calculate the chi-square value, You have to sum up the O minus E squared over E for each category. In question 7, we have the structures of 5 amino acids. You are asked to suggest which amino acids would be most directly converted to pyruvate and oxaloacetate. You can determine this by using the number of carbons in these molecules. Pyruvate has 3 carbons and so does serine. Oxaloacetate and asparagine have 4 carbons. B shows a glycinergic synapse. 1. Describe the differences between a glycinergic synapse and a cholinergic synapse. The differences between their neurotransmitter and receptors have been mentioned in the question, so do not repeat them in your answer. In a cholinergic synapse, sodium ion instead of chloride ions move into the postsynaptic neuron after the binding of neurotransmitters. The diagram also shows that the neurotransmitters re-enter the presynaptic neuron. In the cholinergic synapse, acetylcholine is broken down before the reuptake. B2 suggests why an action potential is less likely to occur after the binding of glycine to receptors. For action potential to occur, the membrane has to be depolarized. The inside of the membrane must become more positive. Chloride ion is negatively charged, so its entrance decreases the membrane potential. When this happens, it will be more difficult to reach the threshold. This will then make it harder to create the action potential. Glycine is an inhibitory neurotransmitter as it decreases the chance and action potential being created in a neuron. 3. Describe how a neuron maintains a resting potential. To maintain a resting potential, a neuron has to keep the insides of its membrane to be more negative than the outside. It is because of the sodium-potassium pumps which actively transport the ions. The transport proteins simultaneously transport 3 sodium ions out and 2 potassium ions into the cell. It is an active movement which requires ATP. There will be leakage through the sodium ion and potassium ion channels too, but there are more potassium ion leaks or diffuses out compared to the sodium ions that enter. Overall, there is a more negative charge inside of the neuron. Remember that you have to mention it is the sodium and potassium ions that move. If you leave out the word ion, you will not be credited. Question 8 wants you to explain how the behavior of the chromosomes in prophase 1 of meiosis results in the appearance shown in figure 8.1. The question only wants you to talk about the behavior of chromosomes, so do not focus on other structures such as the nuclear envelope and the centrosomes. In the photomicrograph, chromosomes are visible. This indicates that chromatin has condensed. Homologous chromosomes pair up to form bivalents in prophase 1 too. Crossing over occurs between the non-sister chromatids. The point of contact between the non-sister chromatids is called the chiasmata. In this process, the exchange of genetic material between the non-sister chromatids occurs. Do not say that they exchange genes, because they have the same gene at the same locus. 9a talks about a disease called PC deficiency. Since the disease is caused by a genetic mutation in only one gene, it is not affected by the environment. Usually, only a polygenic condition is affected by environmental factors. The phenotypes can be categorized into 2 groups, whether you have the disease or don't. There are no intermediates. So, this is a discontinuous variation. B is about coenzymes. You are asked to outline the roles of named coenzymes in aerobic respiration. The coenzymes are NAD, FAD, and coenzyme A. There are 5 marks for this question, so you should talk about all 3 of them. Coenzyme A is involved in the link reaction. It accepts the 2 carbon fragments of pyruvate called the acetyl group. Acetyl-CoA will be formed, and it will go to the Krebs cycle. The 2 carbon fragment will combine with oxaloacetate, forming citrate. This event connects the link reaction to the Krebs cycle. NAD and FAD are reduced to carry hydrogen. When a dehydrogenase enzyme removes hydrogen from a substrate, NAD or FAD will receive it. This is why we call them the coenzyme for dehydrogenase. NAD is being reduced in glycolysis, link reaction, and Krebs cycle. FAD accepts hydrogen in the Krebs cycle only. Reduced NAD and FAD transport the hydrogen they accept to the inner membrane of mitochondria for oxidative phosphorylation to take place. You can also say that they transport hydrogen to the ETC or the cristae of the mitochondria. Question 10 wants you to explain how the homozygous LE genotype results in the dwarf phenotype in sweet peas. The LE gene codes for an enzyme called 3-beta-hydroxylase. The mutated allele, small le, codes for a non-functional version of this enzyme. The amino acid alanine at the active site of the enzyme is replaced with threonine. This causes the active site to no longer be complementary to its substrate, GA20. GA20, an inactive form of gibberellin, cannot be converted by the enzyme to its active form, GA1. There will be less or no active gibberellin to bind to the receptor, GID1, which is found on the cell surface membrane of plant cells. In a normal condition, GA1 should bind to GID1. They will then bind to the DELA protein. Without the binding, there will be no or less gibberellin receptor DELA complexes formed. The complex is required for the breakdown of the DELA protein. So, DELA is not broken down in this case. The protein stays bound to the transcription factors called PIF. PIF cannot exist RNA polymerase to bind to the promoter. If transcription factor and RNA polymerase do not bind to the promoter, genes involved in growth are not switched on. These genes, known as the XET gene, are not transcribed or expressed. The genes must be expressed to stimulate cell elongation and cell division. Without them being switched on, the processes do not take place in the stem. This leads to the production of a dwarf plant.
That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.